The yellow fever mosquito, Aedes aegypti, is a mosquito that can spread the dengue fever, chikungunya, and yellow fever viruses, and other diseases. The mosquito can be recognized by white markings on its legs and a marking in the form of a lyre on the thorax. The mosquito originated in Africa, but is now found in tropical and subtropical regions throughout the world. Spread of disease and prevention. Aedes aegypti is a vector for transmitting several tropical fevers. Only the female bites for blood, which she needs to mature her eggs. To find a host, these mosquitoes are attracted to chemical compounds emitted by mammals. These compounds include ammonia, carbon dioxide, lactic acid, and octanol. Scientists at the Agricultural Research Service have studied the specific chemical structure of octanol to better understand why this chemical attracts the mosquito to its host. They found the mosquito has a preference for right-handed octanol molecules. The yellow fever mosquito can also contribute to the spread of reticulum cell sarcoma among Syrian hamsters. The CDC Travelers page on preventing dengue fever suggests using mosquito repellents that contain DEET. It also suggests, although Aedes aegypti mosquitoes most commonly bite at dusk and dawn, indoors, in shady areas, or when the weather is cloudy, they can bite and spread infection all year long and at any time of day. The mosquitoes prefer to breed in areas of stagnant water, such as flower vases, uncovered barrels, buckets, and discarded tires, but the most dangerous areas are wet shower floors and toilet tanks, as they allow the mosquitoes to breed in their residence. Research has shown that certain chemicals emanating from bacteria in water containers stimulate the female mosquitoes to lay their eggs. They are particularly motivated to lay eggs in water containers that have the correct amounts of specific fatty acids associated with bacteria involved in the degradation of leaves and other organic matter in water. The chemicals associated with the microbial stew are far more stimulating to discerning female mosquitoes than plain or filtered water in which the bacteria once lived. Wear long-sleeved clothing and long trousers when outdoors during the day and evening. Spray permethrin or DEET repellents on clothing, as mosquitoes may bite through thin clothing. Use mosquito netting over the bed if the bedroom is not air-conditioned or screened, and for additional protection, treat the mosquito netting with the insecticide permethrin. Spray permethrin or a similar insecticide in the bedroom before retiring. Mosquito control is currently the best method for disease prevention. This primarily includes source reduction, pesticide spraying or fogging, or the use of mosquito traps like the lethal V-trap. Although the lifespan of an adult Aedes aegypti is 2 to 4 weeks depending on conditions, the eggs can be viable for over a year in a dry state, which allows the mosquito to re-emerge after a cold winter or dry spell. Genetic modification. Aedes aegypti is the subject of investigations that genetically modify the mosquitoes. The modified strain, known as OX513A, requires the antibiotic tetracycline to develop beyond the larval stage and passes this trait on to offspring. Modified males raised in a laboratory develop normally as they are supplied with this chemical and can be released into the wild. However, their subsequent offspring will find no tetracycline in their environments and will never develop into adults. An Oxford firm, Excite, is performing a pilot program in Juazeiro, Brazil, to test the effectiveness of these modifications in reducing disease spread. A 2010 study in the Cayman Islands released of over 3 million OX513A mosquitoes. The wild population of mosquitoes subsequently dropped by 80%. Genomics. The genome of this species of mosquito was sequenced and analyzed by a consortium including scientists at the Institute for Genomic Research, the European Bioinformatics Institute, the Broad Institute, and the University of Notre Dame, and published in 2007. The effort in sequencing its DNA was intended to provide new avenues for research into insecticides and possible genetic modification to prevent the spread of virus. This was the second mosquito species to have its genome sequenced in full. The published data included the 1.38 billion base pairs containing the insect's estimated 15,419 protein-encoding genes. The sequence indicates the species diverged from Drosophila melanogaster about 250 million years ago. and Anopheles gambri. This species diverged about 150 million years ago. Scientific name: the species was first named in 1757 by Frederick Hasselquist in his treatise Eta Parlastenum. Hasselquist was provided with the names and descriptions by his mentor, Carl Linnaeus. This work was later translated into German and published in 1762 as Res nach Palacurrency Steiner. Since the latter is an uncritical reproduction of the former, they are both considered to antedate the starting point for zoological nomenclature in 1758. Nonetheless, the name Aedes aegypti was frequently used, starting with H. G. Dior in 1920. To stabilize the nomenclature, a petition to the International Commission on Zoological Nomenclature was made by P. F. Mattingly, Alan Stone, and Kenneth L. Knight in 1962. It also transpired that, although the name Aedes aegypti was universally used for the yellow fever mosquito, Linnaeus had actually described a species now known as Aedes caspius. In 1964, the Commission ruled in favor of the proposal, validating Linnaeus' name, and transferring it to the species for which it was in general use. The yellow fever mosquito belongs to the tribe Aedini of the dipteran family Calicidae and to the genus Aedes and subgenus Chikungunya. According to one recent analysis, the subgenus Chikungunya of the genus Aedes should be raised to the level of genus. The proposed name change has been ignored by most scientists. At least one scientific journal, the Journal of Medical Entomology, has officially encouraged authors dealing with Aedes mosquitoes to continue to use the traditional names, unless they have particular reasons for not doing so. See also, Mosquito Control, References. External links, Vector Bases Genomic Resource for Aedes aegypti, Aedes aegypti page from University of Sydney, Australia, Aedes aegypti and dengue fever, United States CDC page on dengue fever containing information on prevalence of Aedes aegypti worldwide and past efforts to eradicate it, Aedes aegypti on the UF/AFAS featured creatures website, Walter Reed Hospital distribution, taxonomy, references etc. Excellent image. Aedes aegypti at metapathogen, taxonomy, life cycle, facts, the ecology and biology of Aedes aegypti and Aedes albopictus and the resistant status of Aedes albopictus against organophosphates in Penang, Malaysia.